Hello and welcome to everyone. Today, I am going to share a topic under the subject AQU 3601 Biotechnology Aquaculture by Dr. Nur Asmah. The topic which I'm going to share about is called transgenic fish. So, without further ado, let's get started. I bet you must be wondering what is transgenic fish, right? And how is it work or how it has been done. If you have a look at the picture in the middle, it shows the transgenic process that are done by manually. Transgenic fish is a fish that have one or more DNA sequences from another species have been introduced by artificial means. Animals usually are made transgenic by having a small sequence of foreign DNA injected into a fertilized egg or developing embryo. Plant also can be transgenic by introducing foreign DNA into a variety of different tissue. As for the history of where does transgenic begin take place, it began in China in 1985. The transgenic fishes of many species like common carp, catfish, goldfish, salmon, rainbow trout, and zebrafish have been produced by microinjection of gene coding for rat or human growth hormone. It was found that transgenic fish with human growth hormone gene was twice in size than the non-transgenic fish, thus increasing the meat production. So, what are the types of fish common trait that can be applied to the transgenic process? Transgenic process can help the fish to increase the disease resistance as well as the growth enhancement, can increase the metabolic modification, salinity tolerance, can improve the product for the consumer, and it can help in freeze or cold resistance. As an aquaculturist or fish farmers, what are the goals that we need to achieve when we implement the transgenic process? The answer is we need to create a transgenic organism, which is organism that have genes from two different places from itself or parent or was put by scientists. As you can see in this slide, this is an overview of the procedure and crossing scheme of the fish. The selected embryos has been injected, will be screened, and the positive embryos are raised to adulthood. Then, the fish will be crossbreed with the wild type. This will be resulting in Generation 1 embryos. The Generation 1 embryos will be screened for the expression of the transgene of interest. The first generation embryos are positive for the transgene of interest and then it will raise into adulthood. The independent Generation 1 family should be established to rule out the position effect on the transgene. Each of the family will have one insertion of the transgene of interest. Subsequently, intercross fish within one family to maintain the individual stock. So let's find out about the advantages and disadvantages of using transgenic process. First, the advantages are they are very much like ordinary fish. There are no significant biological differences apart from the fact there have been different number of genes. Second, they can improve health. Genetically modified fish such as salmon contain fatty acids that can help in reducing the risk of heart disease. Third, they can help lower food costs. more food become available without having to rely on wild-caught fish. And finally, they can help alleviate hunger. As for the disadvantages, first, they can outcompete wild-type fish. If the transgenic fish has been released into wild, it can outperform the other wild fish and resulting in total dominance and mess up the natural food chain. Secondly, they may promote allergic reaction. Some fish like salmon that has been modified can increase the allergenicity. Third, they can cause health problems. Some transgenic fish contain high level of growth hormone that may link to the breast or prostate cancer. And finally, they can create a monopoly in the market. The transgenic may have significant difference in meat size or etc. They might be highly demanded and creating a monopoly in market. That's all for me. I hope you enjoy my video as well as gain a new information regarding transgenic fish. Thank you and terima kasih.